Here's what we're going to talk about today. It's a sort of an imposing title, The Black Reaction. But what I'm really going to talk about are two significant major figures in neuroscience. And those two people are Camille Golgi and Santiago Ramon Y Cajal. Very interesting figures in their own way, and then the two of them sort of ended up on a collision course in a certain way, based on the same technology. It's really a kind of an interesting story. So let me start just by giving you an introduction to these two people. So this is Camille Golgi, and you see he's considered a very important person. There's actually a marble statue of him in Pavia, in Italy. And basically, he started out as a physician. He was trained in a physician, and he ended up being involved for a while in psychiatry, very interested with insanity and things of that sort. And there are always little stories. And part of the story here is that while he was working at this hospital, he managed to set aside a little spot to make into a laboratory. And he became very interested in the nervous system. And the idea was that because he was looking at mental illness through his psychiatry interests, he actually had his own idea about what was going on, which was contrary to the general assumption of people's work. And that was, his assumption was that in a certain parallel, if you will, to Virchow, if the mind is sick, it's because the components of the mind are sick. That the cells in the brain, the cells in the nervous system, are the cause, if you will, of disease. So that you would have organic lesions of some sort or other in the neural centers. And so he eventually got very interested in this issue of trying to see structure in the nervous system. At the time he did it, there really weren't very good techniques available. You remember, we've talked a little bit about some of these approaches, but really the nervous system is so complicated as a structure that it's very difficult to make a great deal of sense out of it. I'll give you some examples of what it's like, but it turns out that even when there are staining techniques like hematoxylin, which you're probably all familiar with, it picks up nuclei, or carmine, which is another one that picks up nuclei. Those things don't give you a sense of the spread of the cells themselves. And so this has been a very difficult study for quite a while. It was a difficult study. People tried to do things sort of amazing. You would fix the tissue or treat the tissue, and they were using various kinds of chemical materials to try to harden the tissue, and then actually tease the tissue apart and see if you could see the individual cells that made it up. But of course, when you did that, you broke all of their connections. So it became a very difficult thing to look at. If you did do sections of the tissue, what you would see, and if this is a sort of a simple-minded image of a section, okay, you might see these cellular materials. Those are the dark nuclei, say, that you would see. And then everything else sort of embedded in a kind of a haze. And even if you struggled with the microscope in a lot of ways, you might make out the idea that there was some sort of fibrous structure to it, but it really looked unformed, and it looked quite difficult to make sense out of. Nevertheless, people started suggesting, well, if I tear out a single cell, those single cells that I tear out do start to look as if they have branches. They look, something like this. I always get this, what you would call the soma, and then something sticking out like this. And so one of the ideas that took hold, and we'll get to it a little bit later, is the idea that actually whatever it was that was part of the neuron, so the neuron people did sort of realize had some sorts of projections, that these projections all became part of a major network. And so there was a sort of the idea that the nervous system was like a, not woven, but a complicated, folded network of lots of cells, all of which were connected to each other. So the nuclei were there as indicators of something that nourished this structure, but the nervous system itself was a network. And we'll come to that by the end of this talk. Golgi also got involved in some basic anatomy. He did some very interesting work in kidney structure, and he also became interested in malaria. And so he was the one who actually showed that the plasmodium in malaria, the thing that infects the red cells, right, is a protozoan. And he went through and demonstrated the life cycle of this plasmodium. So it infects the cells, it circulates in the blood in another form, it gets taken up by insects, as you may remember, mosquitoes. And at the same time, he worked on trying to make sense out of the nervous system. And one of the things that happened, it was kind of, you'll see it may have actually been a semi-fortunate accident, was this thing called the black reaction. It's now not called black reaction. I had to get it this way, okay? A lot of people now call it the Golgi Method. And the idea, this is from a memoir by Reticulus. Reticulus was an anatomist in the early 20th century, mostly, that one of his assistants was fixing tissues with potassium dichromate as an agent that would harden the tissues. And accidentally or for whatever reason, incorporated some silver nitrate in the solution. So they put in some silver nitrate, and as a result, they discovered that some of the nerve cells became dramatically enhanced in their appearance. Golgi published this and said, it looks like I can see the nerves independently. And he published it in a paper on the olfactory system. And he drew this kind of diagram of it, where you could actually start to see in his drawing, in his plate, this is an engraved plate, all of these individual cells, which normally you couldn't see. Okay? Well, this is a drawing, and it's a little tricky. This is from 1875, right? One of the documents that I've been looking through on this shows this example of what a slide itself looked like, having been prepared with this material. It's not very helpful. All you see is this sort of smearing material in here, except on the side, it happens to say, this was given to Reticulus as a gift from Golgi in 1886. So a few years after he had developed the technique. If you take that slide and look at it with a microscope, this is what you see. You see, these are the Purkinje cells, and we'll get back to Purkinje again a little later. Here are the nuclei of the Purkinje cells. And what you see going out are all of these fibers, these individual fibers that come out of what appear to be these cells. So they come out and they branch. So the idea is you would have this nuclear structure, and you'd have a process that comes out, and then these things would branch and branch and branch like this. And so you can compare this sort of image, the one that you see here, with this picture, which is what Purkinje himself saw in something like 1830s. Okay? And this is what Purkinje wrote when he described what are now known as Purkinje cells. And sure enough, here are the nuclei. Okay? But no sense at all of the rest of the structure of this cell. Okay? So how does this thing work? It's really kind of an amazing stain. And what it is that, in fact, as they described it here, you first soak the tissue in potassium dichromate for a certain amount of time. And the idea of potassium dichromate at that point was because it's, I guess, a pretty strong oxidizing agent. It started to crystallize or harden the cells. And then you added silver nitrate to it. Silver nitrate, silver has very interesting properties in solution. By now, very few people are aware, as much as they used to be, that it is silver that is the core element of photography, that in the old-fashioned kind of non-digital photography, a paper is impregnated with essentially some form of silver, a silver halide, generally, not nitrate, but a silver halide. And when it's exposed to light, that silver halide in some way becomes unstable. It starts out as small soluble crystals, and then when it's exposed to light, it becomes unstable, and the silver itself emerges as metallic silver from every little spot where there was silver halide. And it's a peculiar kind of thing. It's catalyzed by rather imprecise and random chemistry. It's amazing that it ended up generating a whole field of photography, but it did. Well, something similar must go on in this potassium dichromate embedded tissue when the silver enters it, because what seems to happen is if you have an array of cells, here's one cell, here's another cell, here's another cell, okay. Each one with a nucleus, maybe a small cell here. All of these are soaked in the potassium dichromate and react with it. You then soak this thing in silver nitrate, and what then happens is that only in some of the cells does the silver nitrate get precipitated into what's being described as silver dichromate, silver chromate. And the interesting thing is that not only does it happen here, but the catalysis or the reaction becomes self-perpetuating within the cell. And so what you'll end up with is a single cell that's been stained in the background of all the other material. So as I said, Golgi described this and said, yeah, this is pretty cool. It allowed me to see this material in this way. A number of years later, Santiago Ramon Y Cajal, who we'll talk again a little bit more about, who was, among other things, as well as being a terrific neuroanatomist, was something of a poet and a writer and a painter also by background. He wrote, and this is translated out of the original, but he wrote when he first looked at these reactions He said, what a fantastic sight. On a yellow, completely transparent background, there appear sparsely scattered black fibers, smooth and small or thick and prickly, as well as black triangular and star or rod-shaped bodies, just like fine India ink drawings on transparent Japanese paper. The scientist gazes upon it in astonishment. He's more accustomed to the chaotic images produced by carminic or hematoxylin, which yield one dubious interpretation after another. Here, everything is absolutely clear without any possibility of confusion. There is nothing more to interpret. You just have to look at these guys and see their extensions. They're like plants in the morning frost. I love that phrase. The delighted and astonished gaze cannot tear itself away from this fantastic sight. Methodic wishful thinking has become reality because metal impregnation has produced this magnificent and unexpected slide. Slide meaning microscope slide. Well, that's a bit of enthusiasm. Okay, and between the two of them, between Golgi and Cajal, so let's go back now to the sort of thing that doesn't show up quite as well as you might expect from this, but if you were to see every single cell that's in this area, okay, it would be a complete, in this case, it would be a completely black mess. You wouldn't be able to see anything. But what happens is because only some of the cells have stained, and their processes for each one of them has stained all the way through its entire length, you can actually start to isolate individual cells and think about what's going on. And this is why Golgi was able to draw these kinds of images that showed, for instance, a stellate cell with processes going out in all these directions, or these elongated cells with very thin processes. So you wouldn't be able to see these processes as individuals beforehand. Okay? Things like that, as well as when you get these large cells, now you can see, in addition to their cell bodies, you see the processes that come out from them. Now, Golgi interpreted what he saw here, all these places where these fibers crossed each other and intersected with each other. He interpreted them to be part of what I described earlier as this network. So you have cell bodies, and then you have fibers, you have another cell, and it has fibers. And at this point where the fibers cross over, or if you have a situation where you have one cell and a fiber here, and then another cell here with a fiber this way, you then might argue, in this sort of area, they actually contact each other and make it one continuous cell. The term for this is Syncytium. Okay, and we'll get back to that idea of the nerve reticular syncytium structure in a few minutes. One other thing that showed up in Golgi's studies was that in some of the cells he looked at, or in a number of the cells, he saw an internal reticulum. See this stuff? This type of material here. This complex reticulated, again, it's a reticulum, right? It's a network of material. And he saw it in a case like this where it was much more of a kind of a network throughout the cell, or in this sort of situation as if it was more or less a discrete structure hanging together. This is what we now call the Golgi Body, or the Golgi Apparatus. And up until the 1950s, let's say, late 1950s, this structure was thought to be an artifact of fixation. It was not at all clear that this structure really existed. And if you look at some of the early textbooks, there's a great deal of question about this thing called the Golgi body. And what became even stranger was that as people eventually saw it with the electron microscope, what they saw initially was a stack of membranes. And you probably all remember it from cell structure, that it's this sort of thing. And that doesn't seem to comport very well with the image that's in Golgi's images. Right? So how do we get from one to the other? Well, you may remember, and I don't have it quite as handy as I would like, a fluorescence image that I showed in the cell structure course, in which it was clear that if you have a cell and stain it with a fluorescence image, maybe we'll get back to it when we discuss fluorescence, you can actually see connected bits of fluorescing material that go all the way around the nucleus. And that is still, if it's stained with a Golgi-specific stain, that's still the same structure that Golgi originally described. These images are taken from his Nobel Prize talk, which was in 1906. So he did a very interesting set of observations. He was a major figure in the development of this. Okay, so now to say a few words about Ramon Y Cajal, who was clearly a different sort of person from Golgi. He was apparently much more outgoing and more generally enthusiastic about things. You could tell by the quote I gave you about what he had to say when he finally saw the work of Golgi. Remember, he was working in Barcelona. Golgi was in Italy, and they weren't really in very much contact. Okay? So actually what happened was somebody visited Cajal and said, here's a slide that I got from Golgi. You wouldn't believe what it looks like. Okay? And so the claim of this is that it happened in 1887. Okay? So Cajal was already 32 years old, something like 35 years old, and already doing work in neuroanatomy. But seeing these stains, it opened up an entirely new world for him. And he started looking in detail at the structure of the nervous system in many places. Here are a couple of examples that give you the idea that what he was able to do is look at these sparsely stained tissues and see within them the individual cells that seem to make up the nervous system. So this image of the retina, for instance, to a certain extent, it's the work of an artist. So the artist has said, well, I know these are the photoreceptor cells here. To draw them in a cluster, these are the rods and these would be the cones, okay? We're probably not as beautifully laid out as they are in this image. This is a drawing done from the microscope slide. But what he's saying is that he sees all of these discrete cellular structures. And he seems to indicate And what he's seeing is something that looks like this. Contact area in one case and the contact area in the other case, like this. Okay. With perhaps a space between them. Okay. And so if there's a space between them, then that starts throwing into question this idea of a reticulated nervous system. We'll come to that in another minute. Similarly, you get this sense of this extraordinary range of cell types that are available within the nervous system, different shapes of various sorts. So in the retina, you have these cells, which are the ganglion cells, and then you have amacrine cells, bipolar cells, photoreceptor cells, all of that there. One of the interesting things that Cajal did was he made and it has to be an assumption rather than an observation, that it appears to him that things move in only one direction. They either go out here or in within a single neuron. So that if you draw a neuron, again, a very simple neuron, one end has the elongated part, so we'll extend it a little bit. This would be the axon. And this would be the dendrites here. The idea would be that information or signaling, whatever you want to call it, would only go in one direction, from the dendrites through the body to the axon. And if you look at, for instance, this diagram of the portion of the cortex, he's made an effort to try to draw some of these directional arrows based on what he thinks is going on in the structure of the cells. So the idea is this branched structure of dendrites accepts signals, passes it through the cell body, and then sends it out through this long axon, which then makes contact with other cells. Okay? So that's the sort of thing that he was seeing. Both Cajal and Golgi published enormous books of the details of the anatomy that they saw, going through the entire nervous system of organisms. Cajal had an interesting insight, which was that if you look at embryonic tissues, newborn tissues, rather than adults, the newborn tissues don't yet have myelin developed, or that membrane that covers things, and the stain can be seen much more easily. So there's a way of building in some additional detail into this material. So here we have these two giants. I mean, these books, by the way, I don't have a good way to give you the sense of how detailed and how large they are. They're 500 and 600 pages worth of data, each one, of the details of the nervous system. Cajal's actually is in 3 volumes, and Golgi's is in 1 major volume. Enormous amounts of detail, enormous amounts of work that came through. So, although you know this sort of thing just barely hits the edge of this, I thought I would show you now where this played out. So I've got a lot of text because I thought if you're going to have it, you might as well have it as text. So the idea is that this first idea about how nervous system developed was what I mentioned earlier. This idea, the Reticulus Theory, said that it basically was a continuous syncytial network. Nerve fibers, dendrites, neuronal cells would be directly connected to each other by cytoplasmic bridges. And the bodies were kind of there to provide nourishment. And it turns out this idea that Gerlach had developed in 72 here, okay, was adopted very sort of completely, I don't know the right way to say it, wholeheartedly by Golgi. So Golgi became a really strong proponent of that, and even though he had developed this technique that let you look at an individual cell's process, his interpretation of what he saw was still, here you have a single cell, and it interacts with another cell. And at that point, this contact is through some direct cytoplasmic exchange, so that all these neurons were in fact connected together. At the same time, based on some of the dissection work, some of the morphology work that came out, again, partly because of Golgi's thing, a man by the name of Waldeyer proposed what is now called the Neuron Doctrine. And the Neuron Doctrine is the one that says these cells were discrete. And that the cells were discrete individual cells that, in a certain detail, they started as individual cells in the embryo, they grew to be individual cells in the neural structure. There was no syncytial net of neural cells. The other thing, as I indicated in his diagrams, Cajal also proposed that signals travel only in one direction. Now, what Cajal was able to do, though, was to show in here the idea that what Cajal was seeing was actually a cluster of these processes in which you had the results of one cell and then in some way a target cell this way that then extended back. I've seen a reference to the idea that these images show you synapses. In fact, they don't. What they show is gaps between the end of one neuron and the other, presumably because the stain is incomplete. Okay? So that in fact, there's a problem with this neural model, even though people started accepting it increasingly. Basically, there's still the problem of how the signal is transferred from one neuronal cell to the next. You could see, as I just showed you, that you could get some contact areas that look as if they were specialized. You couldn't imagine how materials would go from one cell to the other unless they actually contacted each other. This has now gotten a lot more complicated, as you may remember from cell structure, because although we didn't really discuss it in detail, what we now have found using the electron microscope, and I'll show you in the next lecture or two about what these things really look like, is that the synapse, the ending of one neural cell, that's cell 1, comes up against the structure, which I'll call cell 2, right? And what seems to happen is small vesicles that you can't see with the light microscope, in fact, are released from one, travel across this little space, and are then taken up by cell 2. So this process of synaptic vesicle release was not available to see by any of these people that were trying to resolve what was going on in the true structure of the nerve system. So there really was an objection you could raise to the idea that cells were distinct. The objection was, well, if they're distinct, how do they talk to each other? How do they interact with each other? Okay? We now know also that a second mechanism of connection, which is the gap junction, again, not visible until we had electron microscopes, was available to see this thing. So, regardless, the work of both of them was sufficiently highly regarded that both Golgi and Ramon Y Cajal were nominated to share the Nobel Prize in physiology or medicine. The process by which the two of them ended up getting the award together involved some very interesting negotiations on the part of the Nobel Prize Committee, much of which is apparently public knowledge or well known, that there were many votes to do this. Originally, Golgi was the only candidate for the award because of the work that he had done. And then some people decided in the committee, felt that in fact Cajal was in many ways, because he was right about the nerve structure, that it was discrete rather than continuous, that he deserved the credit. And so the end result was the Solomon decision. You cut the baby in half, they each get half of the award. So what then happens is a kind of an extraordinary event. And the Nobel Prize, as you know, is awarded as a very elaborate system in Sweden. The king presents the awards. Each of the candidates, each of the recipients then gives a speech about their work. And a number of the things I've presented today come out of the summaries from the Nobel Prize document. But here's what's particularly odd. Okay? Golgi gave his talk on the first day of the ceremony. He gave a long talk, and Cajal gave his talk on the second day. Golgi's talk was a complete and almost vicious description of the value of the reticular theory and how he felt that this model that Cajal had developed with Waldeyer was completely invalid and completely meaningless. So Golgi gave this talk. The next day, Cajal gave his talk in which he showed all of his results that indicated, yeah, to him, these things really are discrete. And so this is one of the kinds of bits of lore that goes on as we go through the history of the development of neuroscience. And there are lots of essays about the relationship between the two men. As it turns out, they allowed this disagreement over what the true nature of the nervous system was to really affect their relationship. And in a certain sense, they really couldn't stand each other. And apparently, Cajal was very seriously affected at the Nobel Prize ceremony when he heard of what Golgi had said. Very sort of a sad note to this. So before I end, I want to make a couple of other comments about Ramon Y Cajal. He was more than just a neuroscientist in a lot of ways. He was known for being extremely left-wing in his politics. But in addition, his mind went in some very interesting ways. He was a painter. He was trained as a painter, and there are paintings that he did. They show up, his skill shows up in a lot of those drawings that you see. He also became a photographer and was particularly interested photography, and his pictures are available if you go to the right sources to see. They're not neuroscience, they're landscapes and things like that. They're very active photographs. He also was a writer of various other kinds of literature. And there's a book that came out a few years ago, a collection of what are called his vacation stories. And they were translated and edited by Laura Offit, who's the woman that wrote the book on Miller's labs and Miller's students. She, a few years earlier, did translations and wrote a lovely introduction to this book called Vacation Stories, which turn out to be science fiction stories. That Cajal's goal was to match the kind of writing of a man now you don't know too much of, named Julius Verne. Yeah, and Verne is the man who wrote Around the World in 80 Days, which you now know as a movie. He also wrote and developed the idea of the Nautilus, of the undersea adventures, which is famous for having an attack of a giant octopus. Anyway, apparently Cajal was particularly impressed by Verne's work, and so he wrote a series of these short, relatively short science fiction stories, and didn't publish them till the very end of his life, because he was afraid that having written these kinds of things would diminish his stature as a scientist. So it's an interesting pairing of two very different individuals who combine to present an enormously deep understanding of the cellular structure of the nervous system. So I think I can stop here.